Recombination is exactly what it sounds like, taking the parts we have and recombining them to make something different. This is how bacteria get genetic variation. Remember that with bacteria, we tend to want to make clones. We want to clone and make something that's exactly the same as what we saw. The problem with that is if we're all exactly the same and something makes one of us sick, it's going to make all of us sick. If one thing kills one of us, it's going to kill all of us. Having the ability to shuffle around some stuff and get new combinations gives you an evolutionary advantage. Horizontal gene transfer is the transfer of genetic information in bacteria to other members of the bacterial population. Notice that it is not offspring. If it was offspring, it would be going from parent to child. So that's vertical. In this case, I've got one bacteria, I've got another bacteria, and they're sharing. They don't even necessarily have to be the same type of bacteria. They don't have to be E. coli and E. coli. It might be Serratia marcescens and E. coli. But the ability to share like that is called horizontal gene transfer. This happens via 3 methods: conjugation, transformation, and transduction. Conjugation uses that pilus that we talked about in chapter 3, I believe, when we talked about the prokaryotic cells. It is a pipe, a hollow tube that can bridge the gap between 2 bacteria. There's going to be a donor and there's going to be a recipient. The donor has to have a plasmid, one of those little circles of DNA that is called a fertility plasmid, in order to form the required pilus. I have to have the genetic code to make one of those things. I have to have the instructions how to make this thing. Transformation. A bacterium takes up DNA floating around. If a bacteria in the environment dies and its DNA kind of gets chopped up and put out into the field that this bacteria is in, it can literally just go and pick up. That is transformation. It's not specific. I may get garbage when I do this, but every now and again when I pick something up, it may be something that is very beneficial to me. Transduction is thanks to bacteriophage. So when I discussed bacteriophage, when we were talking about viruses in chapter 5, I mentioned that sometimes after they insert themselves and they take themselves out, they'll take chunks of the host genome with them and then inject that into the next thing that it infects. Remember that this is a very small genome to begin with. So if I'm taking a chunk of something from somebody else, I'm losing part of my genome. So when I go and I inject that piece of DNA into another cell, I'm transferring information from the first thing I infected, because I took part of its genome, into the next thing that I infected. Depending on what part of my genome as a bacteriophage that I've left behind, I may not be able to do anything to the cell that I just injected myself into. I may have just given them the piece of the genome from the other thing, and because I lost some of my genome, and let's say it was really important for me to be infectious, I don't work anymore as a bacteriophage. So that is transduction. So conjugation involves the exchange of plasmids or other genetic material from a donor going into a recipient via a direct connection. In other words, I actually have to touch a cell, inject my pilus, and transfer over. Plasmids frequently contain information for important characteristics like antibiotic resistance. This is why sometimes we see antibiotic resistance in bacteria that have never been exposed to anything, because somewhere along the line before it became infectious, somebody conjugated with them and shared this resistance gene. Gram-negative cells, conjugation happens as explained. The F factor, also known as the fertility factor, is a plasmid that codes for the conjugation pilus. As I said, I have to have the pilus for this to work at all. In order to make the pilus, I have to have the instructions to do it. F plus will be the donor. They're the ones that actually have the pilus to share the material. F minus will be the recipient. They don't have it yet. The pilus forms on the F plus cell, the fertility positive cell, and links up with the fertility negative cell. They connect and genetic information is transferred. So again, this is gram negative. For gram positive, there's an opening that's created between the two neighboring cells and DNA passes from one to the other. Conjugation is what we call conservative, meaning if I have an antibiotic resistance gene, I'm not just going to give it to you and not keep a copy for myself. I will make a copy and give you the copy, but the original gene still stays with me. I don't give it up. Donors keep a copy of the genetic material and pass on another copy. So that's conjugation. Let's talk about transformation. In transformation, as I said, sometimes bacterial cells die, and when they die, they lyse, they break apart. When they break apart, their genetic material gets leaked. I mean, if I cut you in half, your organs would go out too, right? Same type of thing. When the cell breaks apart, the genetic material is just there. So it goes into the nearby area. The chromosomes break up into fragments, and they're small enough to be accepted by a recipient. It's not the entire genome that's just sitting there. It's literally cut up into small pieces. The genetic material is still viable. It's still readable. It still can work in the cell that picks it up. It's not like because it got cut into pieces, it's all of a sudden useless. As long as there are genes in there that are still intact, it'll work. Transformation is the process of the recipient cell randomly accepting these small fragments of DNA from the environment. The process requires no special cellular equipment, nor does it require a direct contact with another cell. Literally, if that genetic material is lying around, it can just pick it up. Going back to it still being useful, it's like having a used book. The book doesn't become useless because somebody else read it. It's still useful as long as it has all the pages. With the fragments of DNA, as long as there's an entire gene from start to finish, it's useful for the thing that picks it up. It may not be novel, it may not be something new, but it may be something that could, let's say, contain antibiotic resistance. I know I use that a lot, but really and truly that is one of the main things that concerns us as scientists is how quickly antibiotic resistance spreads, because people are getting sick with things that now have no treatment for them because there are no variants that have any susceptibility to any of the antibiotics that we currently have. Tuberculosis is one of those things. In the world today exists tuberculosis that has no treatment because all of the antibiotics that we had no longer work. Now, are all types of tuberculosis like this? No. But there are variants out there that are. And that's scary at this point. So that's transformation. Transduction, bacteriophage are the little viruses that infect bacteria. A bacteriophage serves as an intermediate carrier of genetic material from a donor to a recipient. So literally, this guy is responsible for picking up and dropping off. The cells never really come into contact with each other. We have two types of transduction, generalized and specialized. Generalized means that random fragments of DNA, of disintegrating host DNA, are taken up by the bacteriophage and then transmitted to another bacteria. It can be done pretty much with any gene. Again, if I cut up all of the genetic material inside of a cell that I've infected as a bacteriophage, I can pick any of that up. Specialized, these are highly specific, just like the name suggests. Highly specific genes are regularly incorporated into a bacteriophage. It happens when the phage incorporates into the genome itself. And I'm going to blow these up so that we can look at it better with the actual figures. So the genome incorporates into the host genome as a prophage. Then it's activated and leaves carrying a small segment of the host genome with it. It carries it to another bacterial cell. Let's focusing in on generalized transduction. If you think back to the video where we saw how bacteriophage worked, one of the first things that happened when these activated into bacteriophage that would cause the cell to lyse, they took the genome of the bacteria they infected and cut it up into pieces. Phage infects as normal. And during replication, these chunks of DNA are also floating around. When the phage gets put together, instead of grabbing a piece of the viral DNA, it actually grabs a piece of the host DNA and inputs it into the phage, the capsid. When these go out into the environment to infect other cells, there's this bacteriophage that is not carrying bacteriophage DNA. Instead, it's carrying a chunk of the genome from the original host. And while the actual capsid still does what it does, it injects whatever genetic material is here, this genetic material isn't the viral DNA. So I'm transferring this genetic material from an original host to a new host. Because the virus is defective, as I said, it doesn't have the DNA to make the virus, to make the capsid to do the takeover of the machinery. It's unable to complete the lytic cycle. It doesn't go through that anymore because what I took with me isn't the instructions to do that. The transduced cell survives, and literally whatever the genetic material was that it got, it can use it. It's a used book from another cell, but it can still read the book. Looking at specialized transduction, we talked about the genetic material from the bacteriophage incorporating into the genetics of the bacteria, becoming a prophage. Here is cell A with its prophage incorporated. When this decides to become active and excise itself, cut itself out, instead of it just being that bacteriophage's genome, there's actually a chunk of the host that goes with it. So if you look here, there's red and there's pink now. Pink being the host. So the excised phage DNA contains some of the bacterial DNA. The new viral particles are made. Some contain bacterial DNA in addition to the phage DNA. If you look at all of these phage, they've got red and they've got pink. They don't have just red anymore. As I said before, depending on what you ended up with, it could really affect how these infect bacteria. Cell A, the first cell that we just had our prophage in, lyses and releases the new bacteriophage. The infection with this new genetic material happens, it gets injected, and it gets incorporated into host B's genome, the second cell's genome. Recombination of this genetic material results in two possible outcomes. Either bacterial DNA or a combination of viral and bacterial DNA being incorporated into that bacterial chromosome. So it can literally be this whole chunk here getting incorporated into my chromosome. So you can see pink, red, and purple. Or the only thing that gets incorporated is the genetic material from cell A, which is the pink, and this little circlet here is what's left of the viral DNA. One of the detriments of being a bacteriophage is you have such a small amount of genetic material, as I've said. When you mess with that, leave parts behind, it's not necessarily going to still allow you to be an active bacteriophage. If I don't have the instructions to make a capsid, this isn't going to work. If I don't have the instructions to make the things I need to take over the machinery in the cell, it's not going to work. If I maintain those things, different story. But if I don't, literally all I did was I transferred genes from cell A to cell B, and now I'm useless.